For thousands of years, ancient Egypt has stood in the popular imagination as one of history's most singular, self-contained civilizations, a culture that seemingly sprang up along the Nile and developed almost entirely on its own terms, shielded by deserts on either side from the kind of constant outside migration that shaped so many other ancient societies. Pyramids, pharaohs, hieroglyphics, an entire civilization often pictured as something that grew in splendid isolation, its people, its gods, and its art, appearing fully formed along a single fertile river valley, largely untouched by the churn of migration and conquest that reshaped so many neighboring regions throughout the ancient world. In July of 2025, scientists successfully sequenced the very first complete ancient Egyptian genome ever recovered, and the result quietly overturned that comfortable image, revealing that even in Egypt's earliest chapters, the civilization was already far more genetically connected to the outside world than anyone had definitively proven before. Let's start with understanding exactly why this discovery took so remarkably long to achieve, because Egypt is, in many ways, one of the most extensively studied ancient civilizations on Earth. Egyptologists have spent well over 2 centuries excavating tombs, translating inscriptions, and cataloging artifacts across the Nile Valley. And yet, despite this staggering wealth of archaeological attention, no researcher had ever successfully sequenced a complete ancient Egyptian genome. Not from a pharaoh, not from a commoner, not from anyone. The reason comes down to simple biology and geography. Egypt's hot desert climate is genuinely devastating for the long-term preservation of DNA, which breaks down considerably faster in warm conditions than in the cold, dry environments where most successful ancient genome projects, in places like Siberia or Scandinavia, have traditionally been conducted. Combine that harsh climate with the elaborate mummification practices that altered and often degraded soft tissue, and genetic material even further, and Egypt became something of a graveyard for ancient DNA research specifically, a civilization whose material culture survived brilliantly, while its genetic material simply did not. In 2025, a research team led by scientists at the Francis Crick Institute and Liverpool John Moores University finally succeeded where decades of previous attempts had failed. It's worth noting this was not, strictly speaking, the very first attempt to extract genetic material from ancient Egyptian remains. Back in 2017, a separate research team successfully recovered limited mitochondrial and partial genetic data from a series of mummified remains excavated at Abu Sir el-Malek, a site considerably further north along the Nile, with those samples dating to a period spanning roughly 1,000 to 2,000 years after the Nuwayrat individual's lifetime, stretching from the New Kingdom era through the Roman period of Egyptian history. That earlier research found these later mummies carried ancestry closely resembling ancient populations from the Near East and Eastern Mediterranean, with comparatively less genetic contribution from sub-Saharan Africa than found among many present-day Egyptian populations. However, that earlier study was limited to partial genetic data recovered largely from mitochondrial DNA, the genetic material passed down exclusively through the maternal line, rather than a full genome-wide sequence capable of reflecting a person's entire ancestral makeup. The 2025 Nuwayrat genome represents something considerably more comprehensive and considerably older, the first true, complete genome-wide sequence ever recovered from ancient Egypt, reaching back centuries earlier in Egyptian history than any previous genetic study had managed to achieve. Publishing their findings in the journal Nature, researchers successfully extracted and sequenced a complete genome from the remains of a man whose skeleton had been recovered from a sealed ceramic burial vessel inside a rock-cut tomb at a site called Nuwayrat, located roughly 165 miles south of Cairo. This particular individual lived somewhere between 4,500 and 4,800 years ago, a period spanning the tail end of Egypt's early dynastic era and the beginning of the Old Kingdom, the same broad era that would soon produce the Great Pyramids of Giza. Researchers established this precise dating window using 2 independent methods working in combination: direct radiocarbon, dating of a bone sample recovered from the skeleton itself, cross-checked against careful stylistic analysis of the pottery vessel and surrounding burial context, which matched closely with well-documented early dynastic and early Old Kingdom burial traditions already familiar to Egyptologists from other sites across the region. This convergence between 2 entirely independent dating methods, one purely chemical, the other based on archaeological and art historical comparison, gave researchers genuine confidence in placing this individual's life within such a precisely defined window of early Egyptian history, a level of chronological precision considerably harder to achieve when working with skeletal remains alone, absent this kind of corroborating archaeological context. The key to this genome's remarkable preservation appears to lie precisely in how this man was buried. Rather than being mummified through the elaborate chemical and drying processes Egypt would later become famous for, his body had been placed directly within a large ceramic pot and sealed inside a rock-cut tomb carved into a hillside. Researchers believe this specific combination, burial within a sealed pot inside a naturally cool and stable rock tomb, created unusually favorable conditions for genetic preservation, effectively protecting his skeletal remains, and crucially, the DNA still locked within his petrous bone, the dense portion of the skull located near the inner ear, where genetic material tends to survive considerably longer than in other parts of the body, from the punishing heat and humidity fluctuations that had destroyed genetic material at virtually every other ancient Egyptian site previously examined. Even with these unusually favorable preservation conditions, actually extracting usable, sequenceable genetic material from remains this old required considerable technical skill and patience. Researchers carefully drilled a small sample from deep within the petrous bone under sterile laboratory conditions, specifically designed to prevent modern genetic contamination from researchers' own DNA compromising the ancient sample. The genetic material recovered was still highly fragmented, broken down into short segments typical of DNA that has survived thousands of years, requiring specialized sequencing techniques capable of piecing these fragments back together into a coherent, analyzable genome, rather than the longer, more intact genetic sequences researchers typically work with when studying living individuals. This entire extraction and sequencing process took considerable time and required several independent verification steps before researchers felt confident enough in their results to submit them for publication in a journal as rigorous and highly regarded as Nature. If you find it fascinating how a single burial choice made over 4,000 years ago could determine whether or not modern scientists could ever recover this man's DNA at all, go ahead and hit that like button. It genuinely helps this channel keep bringing you carefully researched deep dives exactly like this one. Before revealing what researchers actually found within this remarkable genome, it's worth pausing to clarify exactly who this man was, and perhaps more importantly, who he was not. Despite popular headlines occasionally suggesting this discovery involved royalty, this individual was almost certainly not a pharaoh, nor a member of Egypt's ruling elite. Skeletal analysis conducted alongside the genetic research revealed considerable wear patterns on his bones, consistent with a lifetime of physically demanding labor, including evidence suggesting he spent long hours seated, potentially indicating he worked as a potter, someone who spent his life shaping clay vessels, not unlike the very pot in which he was eventually laid to rest. Researchers examining his skeleton in detail identified specific markers consistent with this interpretation, including pronounced changes to the muscles and joints of his arms consistent with the repetitive motion of working a rotating potter's wheel, alongside distinctive wear patterns on his hip and knee joints consistent with prolonged sitting on low stools or the ground, a position commonly associated with pottery production throughout ancient Egyptian society based on artistic depictions of potters recovered from tomb paintings and carved reliefs dating to roughly this same period. Osteoarthritis, present in several of his joints, further supports the interpretation of a long working life involving sustained, repetitive physical strain, rather than a life of leisure or purely administrative work that might be expected of someone from Egypt's ruling or scribal classes. He appears to have lived to somewhere between his mid-40s and mid-60s, a genuinely advanced age for this period of history when average life expectancy across ancient Egyptian society is generally estimated to have been considerably shorter, and his relatively modest but respectful tomb burial suggests he held a position of some moderate social standing, without ever approaching anything close to royal or elite status. This distinction matters enormously for understanding the genuine significance of what researchers actually found within his genetic material. This was not evidence about a specific royal bloodline or ruling dynasty. It was evidence about the broader ancestral makeup of ordinary Egyptian society itself during one of the civilization's most formative early periods, precisely the kind of population-level genetic information that carries the most significance for understanding how ancient Egyptian society as a whole actually came together. Now let's examine exactly what this genome revealed. Genetic analysis showed this man's ancestry was overwhelmingly connected to ancient populations of North Africa, accounting for roughly 80% of his overall genetic makeup, closely matching genetic profiles previously recovered from other ancient North African and Neolithic sites. This portion of his ancestry aligned closely with what researchers already expected to find, confirming a genuine, direct genetic continuity between the earliest populations of the Nile Valley and the individuals who would go on to build ancient Egyptian civilization. The remaining 20% of his ancestry, however, is where this discovery becomes genuinely significant. This remaining portion of his genome showed clear genetic connection to ancient populations from the Eastern Fertile Crescent, the region encompassing ancient Mesopotamia, corresponding to parts of modern-day Iraq, and the surrounding territories often referred to collectively as the cradle of civilization. This finding provided the first direct physical genetic confirmation of something historians and archaeologists had already suspected for well over a century, based purely on indirect evidence, but had never before been able to demonstrate through genetic proof. Archaeologists have long documented clear cultural connections between early Egypt and ancient Mesopotamia, similarities in early writing systems, comparable administrative practices, and shared artistic motifs and imagery appearing across both regions during roughly the same historical period. For decades, researchers debated exactly what these cultural similarities actually meant. Did they reflect genuine population movement, people physically migrating between these two ancient centers of civilization? Or did they simply reflect the spread of ideas, trade goods, and cultural practices through indirect exchange and trade networks, absorbed by populations who otherwise remained largely genetically separate from one another? This newly sequenced genome finally provided a direct physical answer, at least for this one specific individual. Cultural exchange between early Egypt and Mesopotamia was accompanied by genuine, detectable population mixing, real people, or at least their recent ancestors, moving between these two regions and contributing directly to the ancestry of the people who would go on to build Egyptian civilization. If uncovering these kinds of definitive genetic answers to century-old archaeological debates fascinates you the way it fascinates me, make sure you're subscribed, because there are many more carefully researched deep dives just like this one still waiting to be explored right here on this channel. This finding carries real significance for understanding precisely how ancient Egyptian civilization actually came into being in the first place. Rather than developing in the kind of splendid, self-contained isolation often depicted in popular imagination, protected entirely by surrounding deserts from meaningful outside contact, the genetic evidence recovered from this single individual suggests early Egyptian society emerged through a genuine blending of populations, a primary North African genetic foundation, enriched and reshaped by real, ongoing contact and intermixing with populations from the Fertile Crescent to the east. This aligns considerably better with what we already understand about ancient trade and migration routes connecting Egypt to the wider ancient Near East, including established overland paths through the Sinai Peninsula and maritime routes connecting Nile Delta ports to the eastern Mediterranean coastline. This finding also invites a useful comparison with what researchers already understand about the genetic makeup of Egyptian populations living today. Present-day Egyptians, extensively studied through numerous modern genetic surveys, generally carry a somewhat more complex ancestral mixture than either this ancient Nuweirat genome or the later Abusir El-Melek mummies described earlier, reflecting many additional centuries of subsequent migration, conquest, and trade following the eras these ancient samples represent, including later population movements connected to the Arab expansion beginning in the 7th century of the Common Era, alongside various waves of Mediterranean, Nubian, and sub-Saharan African contact continuing across the following many centuries. Understanding the deep ancestral starting point represented by this newly sequenced Nuweirat genome, alongside the intermediate genetic snapshot offered by the New Kingdom era Abusir El-Melek mummies, gives researchers considerably more complete chronological reference points for eventually mapping out precisely how Egyptian population genetics gradually shifted and developed across more than 4,000 years of subsequent history, from this earliest formative period all the way through to the present day. It is important to state clearly and precisely what a single genome can and cannot definitively prove, since good science requires exactly this kind of careful qualification. One individual genome, however remarkable and however carefully sequenced, represents a single data point, not necessarily a comprehensive, statistically representative portrait of an entire ancient population spanning millions of people across many centuries. Researchers involved in this study have been appropriately careful to note that confirming exactly how widespread this specific Mesopotamian genetic contribution was across the broader Egyptian population, whether it was common throughout the Nile Valley or more concentrated within specific regions or social groups, will require successfully sequencing additional ancient Egyptian genomes, a task that remains genuinely difficult given how thoroughly Egypt's climate and mummification practices have destroyed genetic material at nearly every other site examined so far. Still, even as a single data point, this genome carries genuine scientific weight, precisely because it represents the very first direct genetic window scientists have ever obtained into this critical, formative period of early Egyptian history. Before this genome was successfully sequenced, every conclusion researchers drew about Egyptian population history during this era rested entirely on indirect evidence, skeletal measurements, cultural artifacts, and comparative archaeology, rather than direct genetic confirmation. This single genome does not answer every remaining question about Egypt's earliest population history, But it does confirm, for the first time through direct genetic evidence, that meaningful population mixing between Egypt and the wider Fertile Crescent region was genuinely occurring during this critical early period, rather than remaining purely speculative. There is another genuinely valuable insight embedded within this same research, connected not to this man's ancestry, but to his skeleton itself and what it reveals about daily life and social structure within early Egyptian society more broadly. The physical evidence of prolonged repetitive strain visible throughout his skeleton, consistent with decades spent working at a potter's wheel, offers researchers a genuinely vivid, individualized window into the working life of an ordinary Egyptian during this formative period, a level of specific, personal detail that historical records alone focused overwhelmingly on pharaohs, elite officials, and religious figures, essentially never preserved for the vast majority of ordinary people who actually made up ancient Egyptian society. This is worth sitting with for a moment, because it represents something genuinely valuable about what ancient DNA research can offer historians beyond simply questions of migration and ancestry. Ancient Egypt's surviving written and artistic record overwhelmingly documents the lives of pharaohs, nobles, and priests, the small elite. Fraction of society whose monuments, tombs, and inscriptions were built to last. Ordinary craftsmen, laborers, and farmers, the people who actually built the pyramids, farmed the fields, and produced the pottery and tools that sustained daily Egyptian life, left behind considerably less direct historical record of their own individual existence. This single genome, and the skeleton it came from, offers researchers a rare, genuinely personal glimpse into exactly this kind of ordinary Egyptian life. A working man, likely a potter, who lived to a genuinely respectable age, was buried with modest care and respect by his community, and carried within his own DNA direct physical evidence of Egypt's deep, genuine connections to the wider ancient world. So when we say scientists were wrong about ancient Egypt, and that new DNA evidence changes everything, here is precisely what that means. The popular image of ancient Egypt as a uniquely isolated civilization, developing largely on its own terms behind protective desert barriers, does not hold up against this new direct genetic evidence. Instead, the very first ancient Egyptian genome ever successfully sequenced reveals a civilization already deeply interconnected with the wider ancient world during its earliest formative centuries. Its people carrying ancestry from both the Nile Valley and the distant Fertile Crescent, physical proof that the cultural exchange historians had already documented through shared writing systems, artistic styles, and trade goods, was accompanied by genuine, real human movement and intermixing between these two ancient centers of civilization. Somewhere within a sealed ceramic pot, inside a quiet rock-cut tomb south of Cairo, one ordinary Egyptian potter's remains waited more than 4,000 years for the right combination of careful burial, favorable preservation conditions, and modern genetic technology to finally reveal his story. He was never a king. He left behind no monument, no inscription, no name that history remembered on its own. And yet, his DNA, patiently recovered and sequenced by modern science, has now told us something genuinely significant about the deep origins of one of history's most celebrated civilizations, a story of connection and exchange, rather than isolation, written not on any monument, but within his own bones.